Hi everyone, welcome back. So last time we explored the process of protein synthesis, DNA transcription, and then RNA translation. We talked a lot about this process, but there are still some things that we haven't really explored. For one, the genes that an organism has are not constantly being transcribed all the time. Organisms don't need to make all of the proteins that they have the potential to make 100% of the time. Some genes have specific and situational uses, and those genes are ones that are only expressed under the right circumstances. As in, they can be activated or deactivated. They can be used to make the mRNA for that gene or not. And they can even, in some cases, be expressed in different levels of quantity. So you can make more of that mRNA or less of that mRNA to then make more or less of the resulting protein. For bacteria, this is particularly important because a bacterium is just a single cell. And that single cell has all of those genes at its disposal. That single cell is responsible for a diversity of jobs, and it needs to only make the materials that are useful for it in the moment that it needs it. And there are different environmental cues that an organism can listen to and observe to help it determine when it needs to express specific genes and not others. For example, some bacteria possess something called the lac operon. This is a set of 3 genes that's responsible for controlling lactose metabolism. We see it in prokaryotes and eukaryotes. And it's something important for any organism that is going to be digesting lactose. But lactose is not something that is guaranteed to occur in an organism's diet. And if lactose is something that is not available for the organism, there's no point to make the enzymes that help break down and metabolize lactose. There's no point to express the lac operon. That would be a really wasteful exercise, especially when resources are very scarce. For a single cell, the importance of using resources wisely is pretty high. And even when lactose is present, but something more easily used like glucose is also present, those lac enzymes are not likely to be produced. That's because glucose is something that is much easier as a resource to utilize rather than lactose. Extra steps are required to make lactose usable for an organism. And so why would an organism want to use lactose, a more challenging resource to use, if glucose is already present? And so bacteria with a lac operon often have to choose when they use the lac operon, when they express those different enzymes. And they choose only to express those enzymes when lactose is present, but other more accessible resources are not present. Another really good example of environmental cues are signals sent by other bacteria. Because bacterial behavior can change a lot depending on if the bacterium is alone or in a group. The bacteria can sense this and change their behavior accordingly. So bacteria will actively be sending signals to one another in the environment, and these signals are something that we call autoinducers. This is the "I'm here too" signal, the "hello neighbor" kind of signal that lets others know that they are not alone. If bacteria sense very few autoinducers, the cell is going to express mostly genes that are necessary for them to use when they are a solitary cell, when they are truly on their own. But if it senses a lot of autoinducers, more than it makes themselves, it's going to switch over to utilize genes and express them that are more useful when the organism is living in a group. Those lifestyles can be very different. Quorum sensing, which is what this is called, is something that plays a pretty major role in a lot of bacterial diseases. Some bacteria can switch on or switch off different virulence strategies when they are more numerous or less numerous. So for example, cholera is a really problematic and significant disease caused by a bacterium called Vibrio cholerae. And this bacterium, when it occurs at low densities, as in when the cells are mostly alone, the cells will produce more virulent or harmful toxins and other proteins. But when they are occurring at high densities, when the cells are not alone, those autoinducers that the cells are sending each other trigger each other to stop making certain virulent compounds, probably to help the cells detach from their environment and leave the host, it's a signal that it might be time for the cells to disperse and try to find new organisms to infect. Staph infections are another really good example of this. Of course, we're familiar with the cause, Staphylococcus aureus. And at low densities, staph is actually less virulent. It does not make certain compounds that it does when it detects that it's at high densities, where those autoinducers are the signal that triggers them to grow more virulent and make these more destructive compounds. Every cell during reproduction is going to have to make sure that all of that DNA is passed on, even if they are not actively using those different genes. Every organism ultimately has to have that full suite of genes, all the different genes that they could potentially need under these different circumstances. So every time a cell divides, it has to make sure that the new cells that it creates all have their own complete copy of the genome. Now, to accomplish this, the cells are going to replicate their genome as they prepare for cell division. This is something that occurs during synthesis phase, or S phase, of interphase before mitosis ever takes place. So in S phase, the goal of a cell is to replicate its entire genome. The genome is ultimately going to be unraveled and exposed, prepared for copying. So the first step in this process is to actively unzip DNA. There is a special enzyme called DNA helicase. It serves kind of like a little zipper fob that will run down the treads of an interlocked zipper. And as it does, it will unravel the molecule. It will break the hydrogen bonds that link the two different strands of that DNA molecule together. Beyond DNA helicase, there's a number of different enzymes that are involved in the process of DNA replication. Ultimately, they all work together to complete that single task of separating the two strands and then making the complementary strand for each of those separate strands. Ultimately, we're going to take one DNA molecule and literally we're just going to unzip it, and then from those two unzipped sides, we're going to build new complete DNA molecules. DNA polymerase is the enzyme that does the building process. This molecule is going to bind to exposed single strands, the things that we call the template strand of DNA, the one that is going to be the reference to build the new strand. As it travels along the strand, it will build nucleotide by nucleotide, finding the complementary match to each nucleotide on the template strand. In the space where this building is taking place, there's going to be a lot of loose nucleotides. It is going to be grabbing nucleotides from the environment and building them one by one by one by one by one. Along the way, it will steadily expand that growing polymer. As it builds, we are taking one single-stranded molecule and turning it into a double-stranded molecule, creating the opposite side. Now, DNA replication is something that happens bidirectionally. It happens on both strands at the same time. Now, just like what we saw with transcription, replication always runs in the same direction from 5' to 3'. That's the building direction. That we will always assemble nucleotides, attaching them to the 3' end of that growing molecule. Now, the replication is something that can happen not just on both strands at the same time, having the amount of time that it takes to do the replication, but it's also something that can happen at multiple locations on the same molecule at once. The result is that we can end up with multiple shorter fragments that form along that new building strand. That will dramatically speed up the amount of time it takes to duplicate the entirety of an organism's genome. Remember, there are many, many genes that form an organism's genome. That's a lot of individual nucleotides that have to be duplicated. Now, once these fragments form, they can later be joined together using another enzyme called DNA ligase. This is the enzyme that will actively link up those smaller fragments on the same strand. It will join them at the backbone so you make one progressively longer and longer and longer strand. This is a really complicated metabolic process. There's a lot of steps that are involved here and many working parts. A lot of enzymes are doing this job cooperatively during replication. Now, when a cell replicates its genome, the intent is to replicate it perfectly, to make a 100% identical copy of that genome, so that the two cells that come from that division are truly identical. However, it's very possible that mistakes can be made. An error in the copy process, when we are replicating all of our genetic information, even something as simple as a single nucleotide being added, subtracted, or swapped out for another, is something that can create a non-identical copy. Two organisms that are created that do not match each other genetically perfectly. This we call a mutation. Any change to the DNA in any form is what we consider to be a mutation. Now, mutations may or may not occur in a gene itself. Not all of an organism's DNA are made up of protein coding genes. There's a lot of stuff that exists in the space between genes. And so the impact of a mutation can vary considerably for a number of reasons. Some mutations are completely neutral, as in they have no meaningful or immediate effect on that organism's ability to survive or reproduce. The majority of mutations fall under this category of being very neutral. Some, however, are quite negative and can be very harmful to an organism that has them. Another small proportion of them are positive, conveying some kind of benefit to that organism. Now, the process of mutation is completely random. Mutations randomly occur periodically. When they occur, how and where is also randomized. The cell does not create mutations intentionally. That is really important to understand. Even though a mutation could potentially be useful, a cell cannot just decide, I'm going to mutate today, and introduce some randomization into its genome. It's not something that a cell is even capable of doing. There are many ways, however, to accidentally create a mutation. Now, mutations that affect the expression of a gene impact either the product of translation. So what does the end protein look like? How does it work? Does it function any differently? Or it can influence the expression of a gene. It can modify how effectively that gene is transcribed and later translated. Is it transcribed more often than it was before or less? In either case, we are modifying the individual nucleotides, which in the case of a gene can directly change the genetic code and alter its meaning. For example, here is a codon that you will find in mRNA. And now if you had a mutation in the original template DNA that created this codon, that swaps this U out for an A, this actually directly changes what amino acid is created from this codon. Now, mutations can come in a lot of different forms. Some of them can have more severe impacts than others, but all of them can be impactful. Substitution mutations are one example. This is an instance where we swap one nucleotide for another. For example, swapping out an A for a G on a strand of DNA. This can cause something we call missense. The resulting amino acid that this codes for changes to a different one. Or it can cause nonsense, as in we could potentially create a stop codon right in the middle of this gene and prematurely end it when it goes to be translated. This can be especially impactful a lot of the time. I should note it's also possible, again, for substitution mutation to have no impact on the resulting protein. That is very possible as well. Mutations can also insert or delete nucleotides from a sequence. That is something we call an indel, as in an insertion deletion. An indel is something that could potentially cause what we call a frame shift, essentially shifting the reading frame of the codons. So if we do that, if we insert something like this T that gets inserted in this example, it's just a single T that gets added, but you can see that every downstream codon of this sequence has been shifted, and now they are all dramatically different from what they were before. A frame shift mutation happens if an indel occurs in a number of inserted or deleted nucleotides that is not divisible by 3. As in, we're not adding or removing whole codons, we're adding or removing like half codons. This will cause the downstream codons to shift. Now, frame shifts can be extremely impactful because they can impact every subsequent codon downstream of the mutation. This can have a really major impact on the resulting protein. It can look wildly different than it did before the mutation, and therefore its function could be wildly different as well. For bacteria, mutations can have a lot of significant impacts in terms of their survival and reproduction. A positive mutation can open a lot of new doors for an organism like a bacterium, allowing it to live in environments that it wasn't really able to do before, or survive on new resources that it wasn't able to do before. Bacteria face a number of different threats from their environments: antimicrobial agents, resource availability, the ability or efficiency of their metabolism relative to their environmental demands, their environmental conditions themselves, and dealing with other microbes too. A random mutation that ends up helping the bacterium could help it tackle one of these significant threats that it faces and help the organism survive and reproduce. And let's not forget that bacteria also have this really amazing ability to do horizontal gene transfer, to move genetic information between individuals that already exist and not just to their descendants. Mutations, therefore, don't have to just stay with the organism that developed it and their immediate descendants. They can be passed around like candy. Bacteria can achieve this in a number of ways, and we've already talked about this in various degrees, but let's cover it again. Number one, we have conjugation. Now, conjugation is when a bacterium will connect to another using a structure called a sex pilus. It's a link that forms between cells, and a plasmid is able to be passed through that hollow connection to the other organism. If that mutation is something that was present on a plasmid, it can be passed between organisms this way. We also have bacterial transformation. So bacteria that die can undergo lysis eventually. They will break apart, and their contents, including their DNA and plasmids, will be released into the environment. And these genomes can begin to break apart into small fragments. Those fragments can be found and picked up by other bacteria and be inserted into their genome. This is a really effective way for bacteria to pick up different things like virulence genes, genes that help an organism cause disease. So this can actually allow a non-pathogenic strain of an organism to shift and gain the ability to cause more significant disease. This is also a mechanism that can be very effective in sharing antibiotic resistance. And then we have transduction. So this occurs only when a bacterium is infected by a bacteriophage, a virus. So bacteriophages have this ability to inject their genetic material into the host's genome. And then when they go to escape that organism and infect another, they have to copy their genome out of that host's genome. In the process, they could accidentally steal or copy a little bit of that bacterial DNA and then insert that into a new host. This can create the opportunity to move bacterial genes between bacterial cells.